All right, let's dive in. Today, we're going to talk about turning that mountain of raw qPCR data into actual, discoveries. We'll take a tour of the CFX Maestro software to see how it can simplify and speed up that whole process, helping you find the biological story hidden in the numbers. Let's get to it. Okay, so picture this. You've spent hours at the bench, your qPCR run finally finishes, and you're staring at a screen just packed with data. It's that moment every single researcher has. Okay, now what? How do you possibly get from all those fluorescence curves to the answer you were looking for? Well, that's exactly where something like CFX Maestro comes in. It's built from the ground up to answer that what now question. The whole idea is simple. Let the software do the complex calculations automatically and quickly with just a few clicks, so you can spend your brain power on what the biology is actually telling you. Okay, so our first step is just to see what happened. We need to get a look at the raw data from each well during the run, and the software gives us a couple of really powerful ways to do that. So the software gives you two essential views. The first is the linear scale, which just shows you the raw fluorescence as it builds up over time. But the real magic happens when you click over to the log scale. This view zooms right in on that crucial exponential phase of the reaction. And that's the part of the curve you absolutely need for accurate CQ calculations. It just gives you a much, much clearer picture. Now, the software is smart. It automatically sets a baseline to calculate those CQ values. But, and this is important, you are always in the driver's seat. If your scientific gut tells you an adjustment is needed, you can literally just click and drag the threshold to where you want it. It's a perfect mix of smart automation and your expert control.
So we've seen the data, right? But before we get too excited and start drawing conclusions, we have to do something critical. Think of it like proofreading your work. We have to make sure the data is actually reliable.
This step is called data quality control, or QC. And really, it's just a series of automated checkpoints. The software scans key parameters, looking for anything that seems unusual or unreliable. This gives you the confidence that your final conclusions are built on a solid, trustworthy foundation. So what's it looking for? First up is reaction efficiency. The sweet spot you're aiming for is between 90 and 110%. Hitting this range confirms that your PCR was working well and that your DNA target was doubling properly with each cycle, which is, well, it's fundamental to getting accurate results. The second big checkpoint is precision. For this, we look at the standard deviation of the CQ values between your technical replicates. Ideally, you want this number to be below 0.2. A low number here is a great sign. It means your pipetting was super consistent and your assay is highly reproducible. Nice work. But let's be real. What happens when your data doesn't hit those perfect numbers? You see an efficiency of 88% or a high standard deviation. It happens to everyone, but it doesn't mean you have to throw out the whole experiment. And this is where the software really helps you out. It makes cleaning up your data so simple. It automatically flags any wells that fail these QC checks for you. Then you literally just click one button, exclude wells, and boom, those questionable data points are grayed out and removed from all the downstream calculations. It's a super fast and transparent way to make sure you're only working with your best data. All right, our data is clean, it's validated. Now we can finally get to the fun part, answering our actual research question about gene expression. And to make this really clear, let's walk through a classic example. We'll analyze a sample study that's comparing gene expression in cancer cells versus normal cells, a type of experiment I'm sure many of you are familiar with. And just like that, the software crunches all the numbers and generates this bar chart. This shows the relative normalized expression, so you can see instantly how the cancer cells compare to your control. Right away, we can spot that genes like ERG and AMCAR are clearly upregulated here. But here's the thing. It's doing so much more than just making a pretty chart. It's running the statistics for you in the background. It automatically performs t-tests to calculate p-values, figuring out if the differences you see are actually statistically significant. You can even set a p-value threshold, say less than 0.05, and the software will pop a little asterisk on the significant results for you. Now, a bar chart is awesome for a quick overview, but sometimes the really juicy insights are hidden in more complex patterns. So let's check out some of the advanced visualization tools that can help us dig a little deeper. I like to think of these different chart types as multiple lenses. Each one lets you look at the exact same data set, but from a totally new angle. And you start to spot things, perspectives, and relationships that you would have completely missed otherwise. First up, the volcano plot. This thing is a lifesaver if you're screening a ton of genes at once. It plots statistical significance against the full change. So the genes that end up in these top corners, bam, those are your VIPs, the ones that are both highly significant and show a massive change in expression. They become your top candidates right away. Next up is the clustergram. This is really cool. It completely reorganizes your data, grouping together genes and samples that have similar expression patterns. Using this color-coded heat map, where red means high expression and green means low, you get this amazing bird's-eye view of the global trends across your entire experiment. So, we've visualized our data from all these different angles. Now for the grand finale, a seriously powerful statistical tool that's designed to answer complex questions with almost zero effort. So, what if your experiment is more complicated than just comparing Group A to Group B? What if you have 3 or 4 or even more biological groups, like different drug treatments or time points? How do you find the significant differences across all of them at once? Well, that's exactly the question that analysis of variance, or ANOVA, is built to answer. And honestly, this is where it just feels like magic. What is traditionally a very complex statistical calculation becomes a single mouse click. In CFX Maestro, you literally select ANOVA, set your p-value threshold, and click Calculate. The software does the rest. It's just unbelievably powerful and easy. So think about it. We went from raw curves to QC, from simple bar charts to advanced clustergrams, and even complex statistics, all in one seamless workflow. It all comes down to this. Your data is holding a story. The real question is, are you using the right tools to uncover it quickly and with confidence? Thanks for watching.